All right, thank you. Good evening, everyone, and what a privilege it is to be able to finish this wonderful evening of science and presentations. So, my name is Jakub. I'm a second-year DPhil with the Pandemic Sciences Institute and, of course, Rubin College. And I will talk a bit about viruses and pandemics in the modern day and what can we, as a not only science but a community, do to stop these from happening. So I'll in particular focus on a particular pathogen called Nipah. Maybe a month ago or so, you may have seen some headlines about a very dangerous, very nasty virus circulating and causing some very harsh disease in Bangladesh and India. What is concerning about this Nipah virus is that we currently don't have any approved drugs or vaccines, and therefore we aren't really protected in case this becomes a public health emergency of international concern or indeed a pandemic. I do my research on Nipah, and it is the sort of main topic of my DPhil. So just to talk a bit about the virus, this is what it looks like here. Here in purple, this is a schematic, but what is concerning is the harsh disease that it causes. So it is we call it a zoonotic spillover infection. So it lives in a population of bats. It is quite happy there. It doesn't cause disease. But then through certain events, for example, contaminating palm sap, which is local delicacy in Bangladesh, people then interact with the virus and become infected. And the disease of Nipah virus is really harsh. You get an incubation period followed by onset of some general symptoms, but then the infection reaches the brain, presenting with really harsh symptoms such as altered mental status, seizures, paralysis, and it is a really nasty pathogen with high mortality. So how did we get to that outbreak? How do we even start a pandemic? Not that we would like to do it intentionally, but we've certainly lived through one, so we are kind of familiar with this. Firstly, I mentioned a reservoir, which is a sort of species. The virus lives there quite happily, doesn't cause disease. Sometimes this virus can jump to another species. Might be human straight off, might not be. In the case of Nipah virus, the first outbreak actually was associated with sort of intense pig farming practices where they put pig farms in forests where the pigs who were not in their natural habitat were interacting with the fruit bats who are the reservoir and became infected. And then the farmers taking care of these pigs became infected through them. In the case of these viruses that we're worried about, then a human is able to infect another human. And this is the sort of point where we start picking these up as a sort of scientific or medical community. We get people that present with these strange symptoms. In this case, really sort of these severe nervous system symptoms. And if this outbreak is serious, people usually then start communicating to the international authorities. And if this is a problem, a public health emergency of international concern is declared, which means basically that this outbreak is serious enough so that multiple countries need to take action. We really need to get to the bottom of this, find out what the infection is, find out what it does, and most importantly, research how to sort of stop it from spreading and ideally treat it, and even more ideally, prevent this next spillover from happening through, for example, vaccination. So I am in a lab that researches these viruses. We do some work on the Ebola, we work on Nipah virus, we work on sort of SARS family of viruses, including SARS-CoV-2. But I really want to stress that the sort of virology that we do, and you'll see some pretty pictures from microscopes that we use later on, is just a small piece of the puzzle, right? So to be able to coordinate this large machinery of pandemic response, we really need to be really effective at communicating with our clinical colleagues, with social scientists, with environmental scientists to stop these spillovers from initially happening, with ethicists to look at, for example, the effects of restrictive measures. It is a sort of large, complex, and I can't emphasize enough how communication and collaboration is important in this. But sort of our small contribution, we can break it into sort of three parts. Firstly, we can look for the viruses before they spill over, right? So we go to, for example, bat roosts. We sample the bats, not using magnifying glasses, but using genetic sequencers, and we look for new viruses, something that we haven't seen before, but is sort of similar to stuff that we've seen before. So, for example, that's how we determined that SARS-CoV-2 is similar to SARS, what we've seen previously. Then we can look at the interplay between the host and pathogen, and this is kind of the bread and butter of my research, where we use various models that we challenge with these viruses and look at this interplay. And it is really kind of like a dance between the host and its immune system and the pathogen. And there's lots of really cool, interesting, evasive mechanisms that the virus is involved to escape from being detected by our immune system, so they can infect more effectively. And then, of course, once we complete that body of knowledge, we can then look into some parts of the virus, whether we can use them as vaccines, what makes a good test, how can we detect this from patient samples. So in the case of Nipah, we are quite well underway. So we, for example, know that these proteins on top of the virus that the virus uses to attach and enter the cells are quite immunogenic, meaning that our immune system can pick them up. And more importantly, we can load them in a vaccine and vaccinate people against it with good immune. We know the receptors, so every virus attaches to a part of a cell, and we can use this for antibody therapeutics. We can also look at immune response, which is sort of my PhD, and we know that where to look for it, so this being the brain in the case of Nipah virus. In practice, what this sort of host pathogen research looks like is it's fundamentally quite simple. We take a model like an animal here. These are sections from lungs of a hamster. This is some spatial transcriptomic output in a brain. And what we can do is look at what the infection looks like, where it is. We can paint the tissue using reagents called antibodies, which we can then light up under a microscope using a laser, and that will sort of illuminate different parts of tissue. This here on the bottom is a technique called spatial transcriptomics, where each dot represents a piece of genetic material, both from the host and from the virus. And we can then turn these data into quantitative outputs, like you can see there on the right. For example, using machine learning models to deconvolute what is a cell here and sort of what markers are present if there's a virus, and what is the distance between this dot and this dot, this being the virus and that being the cell, and how that, for example, impacts signaling pathways. In the case of Nipah virus, some of the work I do personally is we are trying to look at these cells that you can see here in orange that are infected with the Nipah virus and are sort of traveling through blood vessels, therefore potentially bringing the infection and allowing it to spread and cause these very nasty symptoms in the brain. I mentioned models, and one of them being animals. This, of course, we try to avoid as much as possible due to logistical and ethical issues. So we try to develop better models as well as part of virus research that are A, closer to human than mouse, and B, are better in terms of both cost effectiveness and also don't require us to sacrifice lots of animals. One of these is these brain organoids, which are like small brains in a dish. Here you can see 6 of them. They're approximately 2 millimeters across. And we actually grow these out of stem cells. So a few of the speakers here mentioned stem cells. We basically drive them using chemical reactions or chemical reagents, and tell them to grow some parts of the brain, which you can then fuse together, let grow for a bit, and we get this brain. And here you can just see an example of a pathology. This is, for example, a lesion caused by virus, and this is representative of what would you see in an infected human brain. And finally, with a lot of these emerging infections being really, really severe pathogens, they're confined to the highest sort of CL4, we call it, containment level 4 research infrastructure, which is very logistically challenging. So we can also look at, for example, related viruses. If they are sort of similar enough, if we can use them at lower containment, which is cheaper, less resource intensive, and if it gives us the same answer, for example, for testing of some drugs like entry inhibitors, is a way forward. So here we can see on a culture of neurons, so neurons in a dish, these green ones are being infected by a virus which is roughly 80% similar to Nipah virus, but doesn't infect humans. So we can therefore optimize some drug screening, for example, before we test with live Nipah in these expensive assays. So in summary, does this solve the next pandemic? Well, we have to appreciate that these spillovers in pandemics are very, very complex events, and we need large interdisciplinary teams to understand the viruses, their behavior, infection, immunity. We can use these advanced techniques like spatial transcriptomics to look at what's where and how to sort of navigate these complex tissue networks. And this knowledge helps us either design drugs or, better yet, repurpose the drugs we already have. And we are actively working to minimize the requirements for animal models and resource-intensive high containment research for also equity in research, for example. And I just really want to stress that collaboration and data transparency are key in tackling these challenges and getting ahead of future spillover events. So with that, I would like to thank you for listening. I forgot to put a QR code up there, but I am very happy to chat now or look me up on LinkedIn or any other social media platforms. And again, thank you for listening. Thank you for being here. And thank you for being a part of the Rubin community.